everybody. So I would like to give a lecture which is quite interactive. So please stop me in any moment you like. Let's have a discussion together. And I've seen the program of this school, so I'm talking of quite different things with respect to, let's say, neuromorphic computing or treatment of clinical data or simulation of neurons and so on. But the data we are going to obtain here will be the base for that. Okay, it will be the base for neuromorphic computing, knowing the real biological architecture to aspire to. Will be the base for simulation of neural networks. Will be the base to apply machine learning techniques to treat images or to correlate different kind of unstructured data together. So before starting, I would like to know how many of yours have some experience of knowledge of microscopy? So, very few. Okay, good. So I hope I will try and I will succeed in explaining it a little bit more in detail, but in this lecture I also would like to give you examples, okay? So not a tutorial, it's not a tutorial on microscopy, but just to show you which kind of measurement we can perform with those kind of techniques. So the title of my talk is Optical Brain Imaging, but I would like to add a subtitle which explains more in detail what I'm going to talk about. I would like to show you that how it's important to connect the morpho and the functionality in intact neural tissues. Okay. In other words, when we are talking about the brain, it's not possible to disentangle, to detach the information on a single neuron level with the information on a higher and larger area. And it's not possible to connect these two interaction between those areas. So you must provide an imaging tool which is able to have morphology information, functionality information on the whole brain and at the single cell level. So it's a very challenging task. So one first thing and information you're interested to is detect the electrical activity into the brain. And you know there are many clinical approaches to indirectly detect the electrical activity, but the disadvantage that they have is that they work with a very poor resolution. So then when you move to cells and you say, okay, which are the methods for single cell detection of electrical activity? And for people that are working with cell culture, they are using, for example, multi-electrodes, and they are detecting, for example, by using also some voltage-sensitive dyes, and they are detecting, for example, the action potential propagation, the calcium signal propagation. You see here, this is a cute slice of brain. This is not in vivo, a cute slice of brain, and then you detect, you see the wave of propagations of calcium signal. And when you have, for example, the multi-electrode array here, you can place, for example, a cultivated neurons on it, and then every one of these electrodes record electrical activity, but not of a single neuron. Because here you see a neuron can be, the distance between those two is much higher than the dimension of a single cell, and a neuron can be placed also in the middle between those areas. So the advantage of the multi-electrode array, which can be also implanted into the brain, it's that it's very sensitive, but it's limited only to this plane and is not single cell resolution. The same you can say with this detection of voltage sensitive dyes. This is one photon excitation. In other words, you use a lasers which is absorbed by the voltage sensitive dyes. The voltage sensitive dyes, it's sensitive to the local electrical field, and it emits another wavelength in emission, depending the intensity or the shift, depending on the local field, and then you detect with the camera. But you see here that with this one photon emission, again, the sensitivity is very good, but the resolution is not enough for a single molecule, for a single cell, sorry. Now, as you know, probably the possibility to increase the resolution at least in the Z direction, in the axial direction, comes for the confocal microscopy. The confocal microscopy uses an optical filter here, a spatial filter, a pinhole, so that all the photons coming out of the plane are rejected by this pinhole. In fact, as you know from the principle of optics, if the focus is the plane of imaging, it's in the focus of this objective, then the wavefront is plane, and then it's imaged in this pinhole. If the source is outside of the plane, then of course the focus point is nearer, it's closer or far away, and so the light is stopped by this filter. So this filter is like in a photograph camera, it's like the diaphragm, so reduce the depth of field. So if you want to make a portrait, what you do, you just change the diaphragm here, you reduce the depth of field, and if you put in focus only the person you want to focus, and all the trees behind and after they're out of focus. Then in the 1931, Marie Gopper-Meyer, she discovered that it is possible to connect two electronic states in a molecule with two photons. So a two photon excitation. And, but it took about 60 years before that this kind of physical phenomenon was exploited at the microscopy level. And only in the 1990, Dank and Webb, they, for the very first time, they realized the first photon laser scanning microscope. But then this is only updated to 2004, because after that it's not possible anymore to make statistics because of the huge numbers of two-photon microscopes. You see that the technological development of two-photon microscopy was pretty much increasing with an exponential rate. Which is the advantage of two-photon microscopy with respect to one-photon microscopy? First of all, we're talking about laser scanning. That means that in order to produce, for example, that image, I take my laser, I scan my laser all over the image like this, okay? And for every point, I take the number of photons emitted, then I go to the next point, number of photons emitted, and so on. At the end, I reconstruct in my computer the whole image, then I move the objectives to the other plane, another plane, another plane, another plane. At the end, I have a 3D stack of the image. Then, of course, the problem is that when I'm doing this with a confocal microscope, you see this is a cuvette with some fluorescing inside, and I'm focalizing the laser beam in this cuvette. It's a one-photon process, so every molecule absorbs the one photon, and the probability to emit fluorescence is linear with intensity. It's a first-order process. So all the beam excites molecule, you see here, and also here because you don't see because it's absorbed, the laser beam. So all the volume is interested in absorption and emission. Now you are interested only to this focal plane, so you use the famous diaphragm only to visualize the light from this focal plane. And what you do about the light excited here and here is not used. Not only is not used, it's phototoxic, it's photobleaching. So you just bleach the fluorescence of your molecule and you cannot detect anymore. And so the two photon here, it's a second order process. So you have two photon here going from the first excited, from the fundamental level to the first excited level, electronic excited level of the fluorophore. And two molecules have to be excited in parallel, two photons have to be excited in parallel. So here you use a CW, a continuous emitting laser. Here you must use a pulsed lasers, because in order to have the photons, two photons absorbed, you must pack the photons in pulse, in time. So they must arrive all together with an enormous density of energy. So that the probability of absorption of two photons is maximized. Then of course it's a second order process, so it goes like the square of the intensity. So what does it mean? It means here that the beam here is focalized like this, okay? So in this area, the density of photons is lower than in this area because it's not focalized. So here we are under threshold to excite the two-photon process. In a certain moment we go over threshold, and likely the region in which, the volume in which this density of energy is over threshold is only 1 femtoliter. So with the two-photon process you only excite this volume, so you do not need to place any more the optical filter to remove the signal out of the focus, because you do not excite from a priori out of the focus. But then there is another enormous advantage. If you take the brain, for example, and you want an image in the brain in vivo, the problem that you have here is that the brain is a diffusive system. It's a scattering photons. So the photons which are scattered, which are non-ballistic photons, okay, they go out of focus. Okay, so with one photon they excite anyway, because you don't need to have a very high density. And so what you have with one photon, that your beam it's enlarged here, and the resolution of the microscope is exactly the dimension of that excitation volume. So in one photon the excitation volume increases. In two photon, if you enlarge the volume, you lose the power density, you do not excite anymore. And so the threshold of excitation always remains pretty much confined in 1 femtoliter, also with scattering. So what you lose in two photons is just signal, not resolution. But this is easy to recover. You just increase the laser power and then you recover your signal. Of course, something that you cannot neglect is the emission. The emission, in this case, if you have scattering from the objective, is not seen anymore like a single point emitter. So the wavefront here is distorted, and then you lose photons because they come outside of the area of detection. Can this be recovered? There are many techniques, and one of these is adaptive optics. Adaptive optics is a deformable mirror which was invented from astronomy to remove the turbulences of the atmosphere, and can be used also in microscopy, but I won't go much more in detail on this. But if you have questions, if you have anything which is not clear, just stop me. Is it okay? Any question? Okay. Another advantage of two photon is that you are moving from the visible to the near infrared. And here again the scattering length in the infrared is pretty much reduced. So the advantage of two photon is that I can go deeper into the brain, imaging deeper into the brain than one photon, without losing resolution. So how does it work in an apparatus like this? I mean, there is the beam, and then there are the scanning mirror, okay? Then there is the objective. Here you place your mouse, okay, on a stereotaxic holder, okay? And then the light emitted can be detected by a mirror, and then by a dichroic filter separated, because the fluorescence is different in color from the excitation. And then you collect with a photomultiplier, and then you can analyze the fluorescence by spectrometer, and you can trigger your images in vivo with electrocardiogram because the brain is pulsing in vivo. So you want to avoid, for example, movement artifacts. So you must use some tricks to stabilize your images. Another thing which is very interesting is that when you excite with 2 photons, what you do, you go from the fundamental level to the first excited level, then you have intervibrational relaxation, and then you have the emission of fluorescence, okay, which is, in the case of 2 photons, okay, and a shorter wavelength and a higher frequency. But you can have another effect, which is typical in nonlinear optics, in nonlinear crystals. It's the production, it's doubling the frequency by a factor 2. In other words, some part of the tissues, like collagen, for example, the collagen multiply per 2 the frequency. So 2 photons are exactly converted towards a virtual state in a photon which has exactly the double frequency. So this is a coherent process, because the phase of this photon is completely locked to these phases. So it's a coherent process. You keep the phase. The memory of this photon is exactly related to the memory of those photons. Here, this is not the case, because this is an incoherent process, because here the photons remains for a certain times, which is called lifetime of the level, and the phase of this photon is completely random. So what does it mean that the phase of this photon is not random? That you may have some, let's say, coherent effects. You may have that those photons are interfering constructively in second harmonic. And if the second harmonic to chromophore are aligned in the same way, okay, you are adding a strong second harmonic generation signal. So the mouse is operated with a hole into the skull, okay? Then you remove the skull and you implant here a window, and so with some dental glue you fix this window here, and it stays for a month. So you have a month of time to study whatever you want to study. And this is an image, for example, done at a different depth, where you can see it's a transgenic mouse. So first of all, it expresses the green fluorescence protein in neurons, okay? This is a GFP-M transgenic mouse. Only 4% of the neurons are labeled with GFP. Then you can use some Texas red dextran to label the blood vasculature. You see here the blood vasculature, okay? And at the very beginning, now disappear, you see with second harmonic the dura madre, because dura madre is containing collagen, and collagen is producing second harmonic generation signal. Another very big advantage of 2 photon excitation with respect to 1 photon is that the cross-section of absorption of chromophores, of different chromophores, is excited with the same laser. So with the same laser here, you may excite more color. And then with proper filter and different photomultipliers, you may have a multicolor image in one shot. And this is pretty much convenient in many cases. Then, of course, you can exploit the capability of the systems of being very high resolution. And you can zoom in vivo in a single spine. Of course, during the operation, the mouse is gently anesthetized in order to avoid any movement. But some measurements can be done also in vivo with the animal awake. Anyway, let me tell you that here, for example, you can go deep. You can go up to 500, 600, 700, so layer 5, layer 6, and visualize even single spines. and visualize the dimension of the spines. And of course, you are limited in resolution with respect to, let's say, electron microscopy. But the electron microscopy, it's only ex vivo, and the electron microscopy is pretty much time consuming because in order to make a small volume, it takes days and days. So it's a complementary technique, but for in vivo, this is the only opportunity. The resolution here is 1 micron because you have to go deep, and if you go deep, the numerical aperture of the objective cannot be so high like in a pretty much, let's say, thin sample imaging. But 1 micron is enough. 1 micron is enough to see, for example, spinogenesis. And here, for example, it's an image when you place your window on the cerebellum. And the image Purkinje cells. This is a maximum intensity projection, for example, okay, that you have here. It's a stack 3D which is projected on a 2D in order to show you all the stack. And this is an example of 2 colors, as I was telling you before, where Purkinje cell and vasculature are imaged at the same time. Of course, you can use also transgenic mouse that are 2 color labeled. This is a transgenic mouse who has the presynaptic and the postsynaptics labeled differently. So this can be also useful for many kind of studies. Now, let's come back to detect the action potential. Well, I mean, I showed you before that there are already technologies to detect, let's say, calcium imaging of electrical activity in a very superficial way without cellular resolution. Let me show you some new way to detect action potential deep and with single cell and with the single shot action potential temporal resolution. Okay, let's come back to the second harmonic. Do you remember, there are some molecules that can produce second harmonic. If this molecule can be inserted into the membrane, please.
Yeah, I don't think I'm getting the second harmonic thing.
Okay, sure, very good, thank you, I appreciate it. Okay, second harmonic, it's a process in which 2 photons are converted in another photon who has double frequency. So, if 800 nanometer is the wavelength of those 2 photons, this photon has a wavelength of 400 nanometer. So, I absorb in the near infrared and I emit in the blue. This is okay? Good. Now, this is a coherent process. In other words, you see here, this level is exactly the same for those 2. So, those 2 photons are immediately converted in this photon. So, you have 2 sinus waves which are converted in another sinus wave who has double frequency. But the phase of those 2 are exactly locked together. In this case, not. In one photon, this is not the case. Okay? Now, in fluorescence, this is not the case because this is not the double frequency. Because you lose some energy inside here. Okay? Now, those emitters of 2 frequencies are like small antenna. Okay? So, if you see here, suppose there's one is emitter of second harmonic. It's a molecule. It's a molecule that emits second harmonic. And this molecule goes into the membrane of the neuron. So, as an hydrophobic and hydrophilic part. So, if those 2 molecules are emitting second harmonic, if they are absolutely aligned, okay, the 2 signals are exactly the same and you have constructive interference. If they are upside down, the 2 sinus, it's sinus and cosinus, and they are destructive interference. So the amount of interference depends on how those 2 molecules are oriented. Maximum interference, minimum interference, intermediate interference. Okay? Now, these are dipoles. So when you apply an electrical signal, an electrical differential between the membrane, like an action potential, 100 millivolts, for example. These are dipoles. The higher is the field, the more they are aligned. The lower are the field, the more they are oscillating because of thermal noise. So aligned electrical field, no fields oscillating. So second harmonic here it's 5, second harmonics here it's 10. Okay, so the amount of second harmonic here depends on the electrical field across the membrane. Is it more clear? Okay? Here we go. If they are aligned, they are constructive interference. These are aligned, destructive interference. If they are randomly oriented, the signal in second harmonic grows like n, no interference. If they are all oriented, you have the interference, constructive interference, and if this is the beams both sides, the signal in second harmonics grows like the square of the number of molecules, because there is constructive interference. So you immediately, with second harmonic, have oriented structures. You immediately see symmetries, okay? And here we go. If you take your laser, okay, and you scan your laser, instead of scanning all over the beam, all over the cell, you scan here, 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 and here you have a patch clamp, for example. You see here the patch clamp. In red, you have the signal of the patch clamp, and in black, you have the signal of second harmonic. You see how overlapped it is, okay? So in second harmonic, you exactly see what you see with your patch clamp, but without any physical contact. So it's full optical, okay? And this is an enormous advantage, because if you want to visualize more neurons, you cannot use 500. I think that the Guinness, it's 2 or 3 in vivo optical patch clamp, okay? No more. Because it's, also in vivo, can you imagine how difficult it is to make a patch clamp while the animal is moving, for example? Now you see here how nicely, this is a dendrite, how nicely the action potential is going down as you are going away from the soma, for example.
Excuse me?
Please.
But do you visualize the neuron? Or?
Yeah, I visualize the neuron, and you see here that I visualize only the membrane and not the cytoplasm.
So that's a real image you get with this?
Yes, that's a second harmonic generation imaging signal. Yeah.
And for example, if you wanted to stain this neuron?
Yes, this neuron is stained. Exactly, very good question. The neuron can be stained with a bulk loading, or in this case, the pipette is doing a hole. So the pipette is used both for electrical detection and to insert inside of the neuron the chromophores. Exactly. This is the case in vivo, in which you see with this pipette, you patch this neuron, you see the dye is going inside, it's labeling the neuron, and on the soma you are detecting the action potential. Now, of course, you must be fast, because the action potential is 1 millisecond. So if you make an image by making, for example, a scanning with this beam like this, it will take a few hundred milliseconds, and it's gone, because the action potential is only 1 millisecond. So I must image very, very quickly. Then comes the idea of a random access imaging. What is random access imaging? Suppose that I'm interested in this image only to pick 3 objects: the laser, the objective, and the photomultiplier. So after a first image, what do? I jump with my laser beam between this position, and this position, and I neglect all the rest. So in this way, I go much, much faster. And with this kind of techniques, for example, in acute slice, I can imagine more than 1 neurons in parallel. This is, for example, 100 micron depth, single cell resolution, cerebellum. This is acute slice, for example. And here I can image, for example, 5 Purkinje cells, and you see this is a complex spiking behavior. because these are they are excited by climbing fibers, and you know that in the cerebellum, when you have climbing fiber excitation, you have a complex spiking, okay? And you see here that you can study very well synchronizations with very, very high, let's say, precision between those cells. Okay? Now, there is a drawback. Let me be honest. There is a drawback. The signal to noise is not very high. The signal to noise is not very high. And because the second harmonic generation signal for some physical problems is called phase matching, okay? It's produced in the forward direction, not in the backward direction. The energy is conserved in the forward direction. So, but I'm looking in the backward direction. So I'm looking the reflection of the second harmonic back once has been produced, and the signal is very poor. So in order to have that signal to noise, I have to average. And this is possible because it's a trigger excitation. I'm exciting the climbing fiber with the local field, and those action potentials are always there. So I can average. But if I want to see spontaneous activity, then there is a jitter. And if I average, then it's washed out completely. So this kind of second harmonic generation techniques is not good to see spontaneous activity. You see here, in this case, the signal to noise is very, very poor. So if I want to see, I have to record. So how can I move to see spontaneous activity? Well, come back to the scheme here. We say that the second harmonic, it's very interesting, but has the disadvantage of going only one direction. While fluorescence emission is isotropic. In other words, if I excite with 2 photons here and I wait, the probability of emission here, it's the same on all over the solid angle. So if I want to detect in the forward direction, this is much more convenient than second harmonic. Now the problem is that there are not very powerful voltage sensitive dyes with 2 photons. So what we did, together with a group in Connecticut, we developed a new dye, okay, which is very, very powerful in emitting the photons in fluorescence, not anymore in second harmonic. And with this kind of dye, that was possible at that point with a single shot, no averaging, single cell resolution, 200 microsecond temporal resolution to see like you cannot distinguish here if it is an optical signal or a patch clamp signal, but these are 5 cells in parallel. And this is done again in acute slices, okay? And then at that point we can really study the spontaneous activity. Okay, so we have not anymore the problem of averaging the signal. And then we can go, this is again, just to show you the signal to noise is so good that you can go also under pressure phenomenon, for example. And then you can go in vivo. Then you can go in vivo, you make bulk loading, so this voltage sensitive dyes can be loaded just by putting a drop on the dura, and then it's absorbed. It's water soluble dye, and then goes into the membrane, and then always by doing the same random access approach, you can go deep and you can visualize a single neuron action potential in spontaneous activity.
Sir, what's the limitation of number of neurons?
Number of neurons.
Yeah.
That's the main drawback, because this kind of techniques has the advantage with respect to do 1 photon superficial, let's say, detection, has the advantage of going deep and see single cells, but it's limited to a low number of cells. While if you go in the superficial way, okay, you lose the resolution, but you can maybe see 100 and 100 of cells in parallel. So it's a complementary techniques. Can be used if you want to study a few neurons' behavior in some kind of task. It's not useful if you want to, let's say, image entire part of the brain, because I mean, that's not possible in this kind of technique.
Is it possible first to use a more superficial resolution, and then maybe to pick some neurons which would be of interest, and then zoom in and use only these neurons?
Absolutely, yes.
So
That's the way it goes. Exactly. Good. Then, of course, the nice thing is that if you use 1064 pulse laser, you can excite both voltage-sensitive dyes and calcium imaging. And calcium imaging, it's an indirect measurement of the electrical activity, but it's very important. And sometimes they are decoupled. So here is an example. For example, the system is the same, you just make a two-color detection, one for voltage-sensitive dyes and the other one for calcium. And you see here, these are not neurons, these are cardiomyocytes. But you see here that if you measure, instead of measuring different cells, you measure different parts of the cell with this random axis. And you see here that in the reticulum of the tubules inside of the cardiomyocytes, you can detect both action potential and voltage sensitive dyes, and study, for example, the jitter of calcium with respect to the very stable signal of the action potential, and derive a lot of interesting physiological information where, for example, the calcium flux can be completely disentangled, detached from the electrical signal propagation. And this was not known before. So, you know, it's very interesting because you can really go inside the electrical signal propagation. Now, this must be, as I told you at the beginning of my talk, this is not a, let's say, an isolated information. All those information must be related to other two things, plasticity and structure. So in the rest of my talk, I will talk about plasticity and I will talk about structure. Plasticity is very important. I mean, think about one example. Think about Purkinje cells, for example. They are sending an inhibitory signal to the deep cerebellar nuclei. And they must be synchronized, of course, because the motion is somehow driven by this inhibitory signal, and if the inhibitory signal deposing here is not synchronized, you cannot move. So what makes the Purkinje cells synchronize? They receive signals from the climbing fibers coming from the olivary inferior olive, and they are receiving a signal from the parallel fibers. I showed you before an example of complex spiking, okay, induced by parallel fibers. But I could also induce, for example, from climbing, from climbing fibers, generally complex spikings. But I could also induce some simple spikings by parallel fibers. Now, what happens when this system is disrupted? Okay, climbing fibers is one of the few examples in the brain of a system which is able to self-repair after an injury. Okay, so there were examples where, for example, you were damaging with an injection of a chemical compound, you were damaging massively some cells, okay, and some climbing fibers in this case, okay. And then what they saw that there were some sprouting. In other words, there were the generation of new branches trying to reestablish connections. Now the problem here is a massive effect. We want to be precise. We want to study at the single cell level what is happening. So we cannot use a syringe with some, you say, toxic compound. So what we do, we use the same laser which was used to make an image. We use the same laser to cut, to make a damage, to make a dendrotomy. Okay? How does it work? Well, from a physical point of view, if you increase the laser power, you damage, you cook. Okay? So you have a thermal, mechanical, and chemical processes emanating from this. And those kind of processes may be differently. Maybe a nonlinear photoionization, maybe a thermoelastic effects, this to very, very high power. But to moderate power, what we are using, you have a photochemical effect. In other words, you make a one photon process, but instead of going down, or a two photon process, instead of going down, they goes to another state, which is called a triplet state, which is the state over which you have photobleaching and photodamaging in a sample. But these states can have a transition to the single oxygen. It's like a photothermal therapy, for example, okay? Photodynamic therapy. So this singlet oxygen, if the protein, for example, is attached to the chromophore, which absorb the laser light, and which, for example, emit fluorescence, okay, this is the chromophore attached to the protein we want to detect. If the power here is enough, we emit a singlet oxygen. This singlet oxygen has an interaction range of a few nanometers. So the singlet oxygen attaches the protein and inactivates the protein. So it's a knockout, but it's an optical knockout, not a chemical knockout. And with this knockout, you can inhibit protein with an optical signal, but you can also cut dendrites, like here, for example. And in this case, for example, we cut this dendrite. You see, after 1.5 days, this dendrite can be, here we go, can be cut completely after 24 hours. Okay? So with the laser, we can make a laser cut and cut dendrites, or cut even a single spine. You see here, we place the laser on the spine and the neck is still there. So it's very precise. It's much below the diffraction limit of the laser. The resolution here is 1 micron, but we can make a hole of a few tenths of nanometer. So we can study at that point the spinogenesis, and we can study at that point the climbing fiber plasticity. So what we do, we dye inject, we make anterograde tracings, we make the cranial window. So in this case, all the climbing fibers are labeled. Okay, so we label the climbing fibers with virus. Then we make the imaging at a non-surgery. Okay, and then we study after days what was happening. And then you see, for example, here, then if we cut in one area, here, this is the climbing fiber, then we have a sprouted branch. You see here the sprouted branch. The sprouted branch that is coming out from another area. So that means that the climbing fiber is trying to find another connection with the dendrite of the Purkinje cells to establish a new connection. Now the problem is the following. Okay, we see there is this new kind of cable coming out. Okay, but is this cable branching, establishing a synaptic connection to the dendrite or not? And unfortunately, with this 2-photon microscope, we have a resolution of 1 micron, but the synapse is only a few tenths of a nanometer. We don't have resolution to see if the synapse is there or not. So we cannot use 2-photon anymore, microscopy. And now comes the point at the beginning. I want to connect the nanoworld, single spine, to the microworld, single cell, to the mesa world, hippocampus, to the macro world, the whole brain. So I want to make a photo in this room of all of you with the capability to zoom in each one of your cells and show each one of your cells, or show you all together as you are here. So I need to make a photo with the petabyte, okay, with a chip of a petabyte resolution, okay, and with the capability to zoom in until to a single cell. So to do that, I cannot use a single microscopy techniques, but I have to link together different kind of techniques. And then we go at that point to link with electron microscopy. So we have to make two photon correlative electron microscopy. Electron microscopy, it's at that point creating slices and imaging slices and making a 3D stack. So the question is, how can I now connect an in vivo image with an electron microscopy image which is offline, which is ex vivo? So the trick is the following. I take a sprouting branch image, and then with my laser I write an asymmetric figure around this branch. Okay? Now, once I've done it, then I kill the mouse, I remove the brain, I fix the tissue, okay? And then I reconstruct, and there we go. I image the same particular that I was imaging into photon with electron microscopy. And you see how wonderful is the systems, nervous systems. The dendritic, the sprouted branch, which is this one, is exactly adapting to the dendritic arbor of the Purkinje cell. So it's not only a new cable, it's a new intelligent cable which is adapting perfectly to the dendritic arbor to establish new connections. And if you zoom in, you see that all the machinery to create a new synaptic contact in this varicosity, it is present. There are present vesicles, there are present dense core vesicles and other kind of neurotransmitters which are at the base to constitute a new synapse. Are there questions?
Sir, maybe I missed it, but what was the timeline of establishing new connection? How, like after cutting the...
After cutting, after having a marker like this, we remove the brain, we fix the brain, we cut the brain in different slices, we prepare the different slices for electron microscopy imaging, and then with the electron microscopy we recognize this asymmetric figure, and then we zoom in and we find with some image, let's say pattern analysis algorithms, we find exactly the same branch, which is not so easy because it's just like to find a needle in this building, of course. But we develop some techniques which are able, to scale down and to find in all the brain exactly this branch with electron microscopy. So it's a complex procedure.
One thing, so once you have done all this stuff, you get like a stock of photos of electron microscopy.
Yes?
But the slices are, you can also use them, right?
Yes. Of course, this is that point cannot be used with 2 photon anymore, but I mean, that was exactly what we need to understand if there is a contact or not. But you're right, I mean, at least with 2 photon cannot be used anymore because they are prepared for electron microscopy. Yeah. So now we have 2 pieces of the story. We have 2 photon microscopy, which is able to work in vivo, which has the disadvantage of acquiring only superficial layers, arrived to the 5th, 6th layers, cannot go deeper, the 2 photon microscopy, but has the advantage to go in vivo, okay, and having a resolution of 1 micron. The electron microscopy has the advantage of going to higher resolution, but has the disadvantage of being ex vivo, and has disadvantage of analyzing only a very small portion of the volume because it's too much time consuming. Okay? Now, at that point, we need to connect the single neuron to the rest of the brain, because we study these Purkinje cells. We study from the electrophysiological point of view, electrical signal propagation, synchronous, non-synchronous, whatever it is. We study its plasticity, we cut the climbing fiber, we see the sprouting, we see that this sprouting is really establishing a new connection. Good. But those kind of functional skills of that cells depends on where this cell is connected with very long-term projection in another part of the brain. This cell is not isolated into the brain. The behavior of every single cell into the brain depends on the context. And there are different kinds of contexts. There are the very closer context, the cells surrounding that cells. There are the intermediate contexts, the area of the brain where this cell it is. And there are the long-term contexts, where these areas is connected to other areas. So I need a piece of information now. I need to know these kind of cells to the long-term projection, where it is connected. And the problem is that with those two kind of techniques, I cannot do that. Because with 2 photons I can arrive in vivo to the 5th, 6th layer, and with electron microscopy I can image only small parts. Right. So now I have the challenge to move to the whole brain imaging. And the challenging of this whole brain imaging is exactly what I was telling you at the beginning. I mean, I need to make a photo of this class, I need to know where you are seated, but I need to know how also each one of you is composed, cell by cell. So I need a microscopy techniques at that point, which is able to go from single spine, dendritic arbor, whole brain. So how can I accomplish such a kind of scheme? Okay, so I hope you had a nice coffee, and so let's start again. And so, as I was telling you, now the problem is connect the nanoword, the mesa, to the micro. So one possibility is the following. This was developed in the group of Ximi Luo in China. They fixed the brain with a very hard resin, okay? And then with a knife, they make a slice, and during the slice, they make one photon confocal imaging, and then they realign all the stacks together, okay? And they make the whole tomography in this way. So the advantage of this whole brain imaging is that it's high contrast, high resolution. Recently they adapted also to fluorescent staining, because at the beginning it was with Golgi staining, so it was a non-specific. Now they can do with fluorescence. Disadvantage is that it's pretty much time consuming, because you have to slice physically the brain and image, and it takes of course time. And then of course you have to realign all the different pieces. The second techniques use again 2 photons, but in a different approach. You take the brain only fixed, so it's not prepared, it's not in a plastic resin like the other one, it's just fixed, okay? You take, for example, a transgenic mouse, fixed brain, so you make an image of 100 micron, okay, because you can go deep, okay? And then you cut at 50 micron. So you stay away from the border where you cut your imaging. Because on the border are damaged, of course, so it's a problem to stitch together. Okay. So the advantage here is high resolution, high sensitivity. The disadvantage here is that the fixed tissue, it's very dense. With respect to the brain, living brain, you can go in living brain to 800, 700 micron depth. In a fixed tissues, it's very dense, it's very scattering, so you cannot go more than 100 micron. So the experiment they have done so far, in this 100 micron piece, they make only one image in the middle, a 50 micron, because at the very beginning it was damaged by the cut. Going deep, you lose signal because it's scattering. So you just image to the middle. So every 100 micron, okay, you make an image. So you lose 95% of your information, but you can make whole brain imaging. At the Allen Brain Institute in Seattle, for example, they brought, they are Americans, they brought 10 of these microscopes, hundreds of people working on that, like slaves, and of course students. And then they got huge, wonderful images, and they make a wonderful science paper. That's great. But wait a moment. I mean, we are interested to quantitative imaging. One of the goals of the Human Brain Project, in terms of optical imaging, is obtaining quantitative imaging. Measure the density of neurotransmitters in different parts of the brain, measuring the number of cells, measuring the shape of the cells, measuring connections. If you lose 95% of information, you have a nice science paper, which is good, but you don't make quantitative imaging. So this is not suitable. So in order to solve this problem, we make this approach. We use clearing. What does it mean, clearing? We make the brain transparent, okay? So how can you make the brain transparent? Why my hand is not transparent? You know why? Why? Why?
Because you now absorb the photon.
Absorbing, and the second important point, it's scattering. Why it is scattering? You know the Snell law. The Snell law, a spoon in a glass follow two directions because the index of refraction is different. If you make the same index of refraction, you fill all the glass, the spoon is straight. So in order to make the brain transparent, you have to remove the water, who has a different index of refraction with respect to extracellular matrix, and insert the fluid who has the same index of refraction of the extracellular matrix, and then your brain becomes transparent. And there we go. You don't see here, but there is a brain. Okay? But it's transparent. You can see slightly the shadow, like here. This shadow is just the autofluorescence of the neurons which are GFP labeled because of transgenic mouse, because they are transgenic mouse. But you make the brain transparent. And there are different ways to remove water with organic clearing, organic solvents, but each one of those, they have some advantages and disadvantages. Okay? The disadvantages is they all create a tissue shrinkage. So the brain is reduced of a factor 2. And then it's bad. Or it's increased of a factor 5. And this is nice. But I mean, it's bad, I'm joking. It's bad in all cases because you want to preserve the shape of the brain. Okay? You want to preserve the symmetry. The proportion of symmetries, the proportion of sides. And then, of course, to do that, you move to aqueous solvents. The aqueous solvents, they preserve the shape, but they remain clear for a small amount of time. So, the problem is, first of all, this kind of stuff, of clearing, either they change the dimension of the brain. Either they are not very transparent, either they are transparent but they do not remain transparent for a long time. But there is four issues which is very important. This is only for clearing brain which have been already labeled in advance, which means transgenic mouse. But if I want to make post-mortem labeling, this is what we want to do, because we want to label different structure in partner. We cannot do like this. So recently, last year, they invented a new method which is called Clarity, which is a completely different philosophy. Clarity removes lipids completely and creates a sort of hydrogel. The brain becomes a sort of hydrogel. But the nice thing is everything is fixed. All the structure remains like they are. And the nice thing is that if it is an hydrogel, then the antibody can penetrate inside. Okay? Can penetrate inside and can label post-mortem the structure you are interested in. So it's and unfortunately, Clarity use the final step of Clarity is the clearing, let's say, protocol, because Clarity first remove the lipids and then clear the brain. The clearing the brain, it's done with a compound which is called Focus Clear, and a lot of companies done a lot of money, like Olympus have done an objective exactly for Focus Clear, Zeiss have done an objective exactly for Focus Clear. Unfortunately, Focus Clear costs about 500 euros per brain. Okay, it's very, very expensive, and sometimes the dimension of the brain can augment up to a factor 2. So we set up a new kind of clearing, which is not new, but was new for the very first time in brain. This was used for single cell, and we use it for brain, which is called thiodietanol, TDE. And TDE is able to clear the brain and can be used also with Clarity, okay? With Clarity protocol. And the nice thing is that if you use the other Clarity, they can take 1 week to clear the brain, while TDE is only 6 hours. But the nice thing is that it costs 200 times less than Focus Clear, because this is a very, very common compound. And that's the nice news with the two photon serial section. Do you remember I showed you at the beginning the 2 photon serial section? Do you remember I told you you take only one plane in 100 micron because of scattering? And now with this clearing you can go up to 2, 3, 8 millimeter depending on the objective you are using. You can match the objective with the index of refraction, and you can go deep without losing no one cells. You have 100% of information up to deep, which is just limited by the penetration depth of the objectives, and you don't physically slice so many times every 100 microns. You slice every 8 millimeters. So, for example, the whole brain of a mouse can be sliced only 2, 3 times maximum, no more. And as I was telling you, we can apply TDE. This was a measurement made with TDE in the transgenic mouse with let's say pre-labeled, because it's transgenic GFP. But we can use TDE also with Clarity, and the nice news here is that we can make post-mortem labeling. So let me show you an example of the serial two-photon sectioning performed with Clarity and TDE. This is a post-mortem labeling. You see here we are going down the different planes, okay? Okay. And what you see here, color, it's because we label with anti-GFP IgG Alexa fluor, okay? Which is labeling the neurons, GFP, and it's going very deep. So it is possible to have big pieces, let's say 1 millimeter of brain, all intact, and all post-mortem labeling with the antibody we are interested in. This is, for example, another measurements. This is performed with SET ground, and this is a postsynaptic density, 95. The postsynaptic density receptors, what they do is they establish a class of receptors nearby the synapse, and the number of receptors is directly correlated with the strength of the synapse, the strength of the electrical connection. So having the capability to have a quantitative imaging of the number of synapses in the whole brain, this is an important piece of information, okay? So now, what you can see here, in this case, is 95, postsynaptic density 95. What it's doing here, it's creating a scaffolding for the other receptor proteins, okay? And it is, at that point, as an importance, pretty much importance in establishing, let me show you again the movie, in establishing, let's say, a certain distribution into the brain. Or you can image the entire hippocampus, for example. This is the hippocampus image on a transgenic mouse, GFPM, labeled with TDE, and you can see here, this is a part of the hippocampus, both the cell, both the fiber connections, or you can state the human brain. This is human brain, stay post-mortem, of course, because it cannot be done in a transgenic way. And here we have a two-color labeling. This is clarity on human brain with TDE, and we have DAPI in green, and parvabumin is red on processes here. And here on human brain, this is an example of two colors of human brain. But the nice thing here is that you can make a multi-route. So you can make a first, you decide to make of that piece of brain a map of all the nucleus and a map of all the processes. Then you remove that antibody and you label the same piece with other antibodies many times. And this is the second round, for example, where we use antibody anti-GFAP, which is labeling glial cell, in this case astrocytes, and you see you superimpose to the DAPI the astrocytes labeling, then you remove this labeling and you label other things, and then what you do is you make a multi-level maps. So you make a maps of connection, and maps of transcriptions, and maps of cell distribution, and maps of neurotransmitters, and so on and so on. A multicolor level maps. And that's what you need, of course. Because a single maps of cell doesn't mean anything if you don't have a maps of connection, if you don't have a maps of neurotransmitters, if you don't have a maps of, for example, other kind of genetic expression, and so on. Then, of course, the disadvantage of these techniques is that it's faster, of course, than other kind of techniques. Okay, and let me spend some words with respect to electron microscopy. Electron microscopy is an exactly complementary techniques. Let me tell you that it doesn't exist the killer application. It does not exist. Okay, so every kind of techniques has advantages and disadvantages. Okay? The electron microscopy, there are many groups in the world, like the Denk group, the group of Javier de Felipe in Spain, and many other groups, the group of Lichten in the United States, and many other groups in the world. They are working on that. The approach is slicing piece of the brain and making electron microscopy, which has the enormous advantage of being a much higher resolution, but has the disadvantage of being time consuming. So to make the whole brain with electron microscopy, it takes years. Okay, so it's not possible. But on the other side, the approach could be use all the techniques altogether, using electron microscopy when you want to zoom in some areas you are interested in, using 2-photon microscopy serial to have a much larger volume connection with high sensitivity multicolor. But I mean, to make the whole hippocampus, for example, it can take 24 hours or even more times in imaging. Okay, only the hippocampus. So maybe sometimes if you want to make higher resolution, it takes a few days also. But if you want to make the whole brain, even with 2-photon microscopy, it takes 1 month. Okay, so it's too much. Okay, so you need another techniques, which maybe has the same resolution of 2-photon microscopy. Please.
I don't understand why it's too much. Like making the whole brain, like having the map of the whole brain in 2 months. Why it's too much?
Because to make an image in 2-photon, you have to make the single beam going like this. And every point you have to stay a certain time to collect photons.
Yeah, I understand that how like it takes to be too much, but 2 months. But like, don't understand why it's too much. Like you have the map after 2 months. So
you have 2 months. No, I don't say 2 months. I say that 20 hours can be, 24 hours can be for hippocampus. If you want to make the whole brain, 2 months is the whole process, imaging, stitching, and all the stuff all together. But the imaging can be 1 week, for example, for the whole brain with 2-photon microscopy. 1 month is the whole process. I will describe later why you need 1 month. But the pure optical imaging can be 1 week for the whole brain with 2 photons, because you need to move the beam like this, okay? Which is much faster of 1 year with electron microscopy, okay? But it is anyway 1 week, which is not very, very fast, let's say.
Yeah, but when you map the whole brain,
yes,
via whatever process, afterwards you have the map of the brain. So now why there is that much attempt to make a new method to be faster? I mean, even if it takes 1 year, you can map the whole brain in 1 year, and after that you can have the atlas of the brain, maybe.
This is not true, because there is the biological variability.
Yeah.
You have to make 100 of brains. If you take the paper of Allen Brain Institute, they make about 450 brains to make a good statistics. So you need to be fast. Otherwise, it's just a proof of principle, but you can accumulate data. In the Human Brain Project, we are going to accumulate 100 and 100 of brains. We cannot accumulate only 5 brains in 5 years. That's the point. There was another question there?
No, it was related. So, how long can a slice stay like without deteriorating or whatever, while it is processed?
No, this is not a problem.
Okay.
A slice can be imaged many times, so can stay for a month.
And how many devices do you have if you would like to process many brains in parallel? Could you do that?
Well, at the moment we have, I will show you, we have about 2 devices in 2-photon and 2 devices in this other modality I'm going to show you now. Yeah. Well, if I want to go faster, I have to use the light sheet microscopy. Light sheet microscopy works on this principle. You have the whole brain. The whole brain is illuminated with a light sheet of microscopy like this. which is illuminating a portion of the brain. And you look from these sides. Then you move the light sheet like this, and you rotate the brain, like the computer-assisted tomography, exactly the same principle. And then you make the 3D, but this is much faster, because you don't illuminate one point, you illuminate a good set of points in one shot. And you don't use a photomultiplier, you use a camera. And this is, the advantage is you can image one entire brain in 1 day, and not 1 week. So it's 7 times faster. It's non-destructive. You don't have to cut the brain. The brain is entire. Okay? I come. You have an intrinsic optical sectioning. Of course, you need the brain cleared. And then you need, of course, to clear well the brain. Please.
If you say that the imaging takes then 1 day, 1 week, whatever, does it mean that after this time you already have the data and the way you could use it? Or is still a pipeline of image processing?
I'm going to
Okay.
answer you in the next
Because I know
slides.
from polarized light imaging from Pascal Amos that
Yeah.
this part takes the major.
I am going to answer you. Exactly. This is the system we have developed, which is totally new, because again, the system is not totally transparent. So you have scattering. So you have to remove the scattering. So what we do, we implemented the confocal rejection with the light sheet. So in our system, okay, the light sheet was done with this beam which was scanned in this direction. Here in this chamber is the brain which is rotated, and this every one of these positions is synchronized with this mirror in one slit here. So this is a pivot point, a fixed point. This is a confocal slit. So you remove the scattering in this way. So even if the brain is not totally transparent, you can remove the scattering. And this is, we call this confocal light sheet microscopy. And with this second mirror, we recreate the image on this detector. And here you see, if you go very well deep into the brain, this is whole brain, this is not cut. And you want to image very dense processes. With the simple light sheet microscopy, you have scattering, you have blurring of images. As soon as you use the slit, you lose signal, it is true, but then you are going to gain in terms of contrast. And then you can see and solve single processes. And so you can reconstruct the entire brain. This is about 10 terabyte size data. This is, for example, cerebellum. But the nice thing is that you can zoom in one region, okay? And then you can try to follow, for example, processes in one area of the brain and track processes, even millimeters processes, from one area to another area of the brain. And then if you use clarity, for example, you see how you can visualize processes and follow processes all over the brain. Of course, then comes all the processing you were telling before. And that takes another 2 weeks. Because then you have hundreds or thousands of stacks, and you have to stitch everything together. So we have a fiber optics from our center in Florence to Cineca and to Zurich. Okay? So we transfer our data to the supercomputer with these dedicated fibers, and then in remote, okay, so from people from outside, they upload their instruments to visualize data. And these are instruments based on machine learning, for example, because you have to stack automatically, you cannot do manually. There are 10 terabytes data. If you think about the human brain, it's 10 petabytes data. It's impossible to do by and petabyte flops of petaflops of power. So you can do by manually, and then you have to navigate and stitch and navigate. We developed, for example, a Google Brain software, which is inspired to Street View. It's used exactly the same algorithms of Street View, okay? And then you can navigate into the brain, you can click twice, change the direction, change the navigation path, and so on. And this is a remote can be accessed by everyone in Cineca in Bologna, for example. And we develop systems to count automatically the number of cells. So someone was asking me about Purkinje cells from Spain. Who was the guy? Here we go. So here it's a system that counts cell by cell in the Purkinje in the cerebellum and tells you for every cell's X, Y, Z position with an error of 3%. Okay? So here we have about 222 cells in this area, for example. Okay? So if you want, for example, detect the position of the cells in autism, because autism is going to alter it, like Huntington disease, autism, schizophrenia, obsessive compulsion, it's going to alter it. The position of the Purkinje cell into the cerebellum, you can use this kind of tools to say that. And then, of course, you can use other things like transgenic mouse that during an activity, when they are exposed for 1 hour to an activity, the cells which are activated, they become fluorescent. Then you sacrifice the mouse after 4 hours, and then you make a map, like an fMRI map. It's an ex vivo, of course, but with a voxel resolution 1 billion times higher. The resolution, 1 billion times than fMRI. So it opens up completely a new field in determining, let's say, on the whole brain, the functional maps. Or you can make a vasculature architecture. And this is also important because you can make multicolor. I don't have time to describe the second generation apparatus we are building, but the second generation apparatus has multicolor excitation, about 5-6 lasers. So you can make in the same map 5-6 antibody labelings and then relabeled with other 5-6. So let's say in a map, in a month, you can make a 10 level, 15 level maps of the brain. So now, just 2 minutes of my talk, I want to finish just connecting. Do you remember we were doing plasticity of a single neuron in vivo? Now we want to see this single neuron, where this single neuron is projecting. So we correlate 2 photons with electron microscopy. Now we are correlating 2 photons with light sheet microscopy. So what we do, we make 2 photon imaging, we make tissue excision, we clear the brain, and we make light sheet imaging. But we must find exactly the same field in light sheet as the 2 photon field we were observing before, exactly as we did with electron microscopy. So, unfortunately, the contrast is not very high, but here we have some shadows, and the shadows are the blood vessels. Okay? So, in a 2 photon imaging, we have the pattern of the blood vessels. Then we illuminate with green light. And with green light, we have a very high contrast on the blood vessels. So we can overlap the 2 photons with the wide field green light. When we sacrifice the mouse, we clear the brain, and when we clear the brain, all the buffer goes in all the parts of the brain except the vessels. Here, in light sheet microscopy, we have some residual fluorescence, which is called unavoidable effects, which is a bad effect. For us, it's very good because it gives the fingerprint of the blood vasculature, which is present in 2 photons, in wide field, and in light sheet. So we can overlap the images. And this is the image in 2 photons, this is the image in light sheet, so we can find exactly the same image. And then we can place this image in the context of the whole brain. And here we go. This is the image we acquire into photon, okay, which is placed in the context of the whole brain, okay. So we know where this projection is going to 1 centimeter direction, so 1 centimeter distance. And so we can close the circle of the physiology of that single neuron by knowing the projection of that neuron to very long distance. So let me close here. I know there are a lot of things, I know that I cannot go deep into details, but let me say that this kind of lecture was just to give you a flavor of what we can do with some. I don't want to, of course, don't want to pretend to talk of all optical imaging because it's much vaster than this, but the take-home messages is the following. When you are talking about the brain, it's always think about the multimodal approach. You always need to connect the single neuron with the whole context. So, I would like just to be grateful to all these people coming from all over the world which took part to all those kind of measurements, and let me also thank you for your attention.